Hello, good morning, class. Let's get started. Today we're going to be doing lecture 8, which is about heredity. So we'll go through some of the basic theory behind um, heredity, and then the second half of the lecture we are going to be solving genetic problems. To understand heredity, we have to look at the process of creating gametes. Gametes are sperms or egg, and the process for creating them is what we call meiosis. Right? Meiosis. So we're gonna compare and contrast the similarity and differences between meiosis and mitosis. The purpose of meiosis is for sexual reproduction, in which there is a fusion between reproductive cells from two parents. So in humans, in animals, we have the fusion of the sperm and the egg, right? In plants, we have the fusion of pollen and the egg. So these are called reproductive cells, or we can call them gametes. Gametes are reproductive cells. So to produce these gametes, we would need to undergo the process of meiosis, where the number of chromosomes are going to be reduced by half. Humans have 46 chromosomes, so in the gametes, we would have 23 chromosomes in the egg and 23 chromosomes in the sperm, and when they join together, 23 gives you back the 46, right? So the process of meiosis is the basis of sexual reproduction. Now, just to remind you, previously we learned about asexual reproduction, and that only requires one individual. There's no genetic variety in it, and it's just done through mitosis, right? And here, meiosis would be the opposite, right? It's the process that enables sexual reproduction to occur. Humans are diploid organisms. Diploid means there are two sets of chromosomes in each of the somatic cells. Okay? In case you forget, somatic cells are just bodily cells. Cells that are not sperm or egg, we call them somatic cells. It could be liver cells, could be brain cells, could be skin cell. All these are considered as somatic cells, and each of them would have two sets of chromosomes. Right? That's why we are called diploid. So when the when the body produces the gamete, the sperm and the egg, as I mentioned already, because the number of chromosomes is half, they only have one set of chromosome, and as such, we call them haploid. Okay, so here we have a haploid egg, which we just write the letter N. N is the number of chromosomes. Number of chromosomes in one set. Okay, so for humans, N is 23. So if you take 23, they fertilize each other, and you end up having a 2N, a diploid cell. A diploid cell, 46 now. Okay. Other set numbers are possible. If one set has 3 chromosomes here, a diploid would be 6 chromosomes in total, right? 2 sets. Triploid means 3 sets. So, 3 of each, 3 of red, 3 of purple, and 3 of yellow. And tetraploid would means 4 sets. Okay? In fruits and plants, polyploidy is common. Right? This is multiple sets of chromosomes. Okay? So we have like strawberry is 8N, octaploid, and this is a sweet potato or yam. That is a hexaploid, okay? 6N. So polyploidy just means multiple sets of chromosomes. Right?
Morning, sir.
Morning. So meiosis is only possible for organisms with even sets of chromosomes. Okay? Because in meiosis, we are going to divide the genetic material by half, right? So if something is triploid, for example, is not possible to produce gamete because 3 divided by 2 is 1.5. You cannot have 1.5 of a set in each of the gametes. Okay? So it has to be even number sets of chromosomes for it to be capable of producing gametes, okay? For it to be capable of sexual reproduction. I'm not sure if you ever wonder how is it possible to have something like a seedless watermelon. If there is no seeds, then how do you grow the watermelon, right? It turns out seedless watermelon is a polyploid fruit. That means it has a multiple set of chromosomes, right? So regular watermelon, the ones with the seeds, with the black seeds, those are actually diploid. And then they have a special technique of breeding them to create something that's a tetraploid, so 4 sets. Okay. So if you breed if you take the regular watermelon, the 2N, and you cross it with the specially created 4N, what happens is you end up creating 3N, sterile seedless watermelon. Okay? Remember, if you have odd number of chromosome sets, it's sterile, right? You're not going to have any seeds. You're not going to be able to produce any gametes, and that's what the seeds are, right? The seeds are essentially what allows the plant to propagate. And if you're not able to do that, then it becomes seedless. So the process of fertilization involves the sperm fertilizing the egg. And once that happens, what you get is a zygote. Right? The zygote is a single cell that you get with full sets with all the chromosomes, two full sets of chromosomes, and then it will rapidly divide as soon as it's being formed, and eventually it is going to become the embryo, okay, which of course is the baby, right? So zygote is just the immediate cell that is formed when a sperm fertilizes the egg, and that becomes the baby eventually. So I'm just going to read this to you and explain some of the terms here. The process of sexual reproduction begins with meiosis in the germ cells that are found in the gonads. So what are germ cells? Germ cells are cells that are going to become the sperm and the egg. Okay, so these will become, we can say better, that they will undergo meiosis to become sperm or egg. Okay. The germ cells are the cells that are found in the reproductive organs. They are found in the testicles for male or the ovaries for females. And once they undergo meiosis, the gametes that they produce are going to be haploid. So you start off with 2N. This is the germ cell. And then you will basically produce 4 haploid cells. These are gametes. So that's one difference, right, between mitosis and meiosis. Mitosis creates 2 cells. Meiosis creates 4 cells. And we're going to take a closer look at this process. And again, you should know humans are not the only ones who are doing this, right? Plants also can do sexual reproduction. But plants are a little bit different because they have both male and female parts. That's why plants can self-fertilize. The pollens are found on the anther, and then the egg is found in the ovary. If you grow plants in your garden, right, or grow them indoor, and you want to fertilize them, you can pick up the pollen with like a Q-tip or something, and then you brush it in the centerpiece of the flower, right? And that's going to cause the pollen to go in and fertilize the egg. And the result is that you will grow the fruit, right? So like tomatoes, for example. Okay, so here is an overview of meiosis. We start with two sets of chromosomes, the red and the blue here. We call that 2N. And we're going to copy all the genetic information. This is just DNA replication. And afterwards, you are going to have, it's still 2N because you only have two sets, right? Remember, if they're stuck together, is count as one chromosome. So that's like, even though you duplicate them because they're still connected at the centromere, it's still considered as one. And these are sister chromatids, of course. Then you have the first division, meiosis one. So you separate them, and now you only have one set and one set. And then you're going to divide again, and you end up having four gametes. So I'm going to draw this out for you to help you understand it a little bit better. If you have colors, please take them out. You will need to have three colors, preferably black, red, and blue. Okay, so this is meiosis. So this would be a a germ cell. Germ cells are diploid, 2N, and they are found in reproductive organs, the gonads, right? The gonads. For simplicity, we are just going to be looking at two sets of chromosomes. Okay. But remember, we have 23 pairs, right? But again, just to keep things simple, we will only be looking at two of them, like this. Right, so this is like chromosome 1 and 1 over here, chromosome 2 and 2 over here. And again, in the actual cell, you would have 3, 3, 4, 4, 5, 5, all the way to 23, 23, right? So I just want to make sure that you remember some of the terminologies. What's the relationship between these two? Does anybody remember? Starts with an H.
I don't remember, sir.
Okay, thank you. Anyone else? That's quite all right. So this is homologous. Right? Homologous, which means they are same length, same size, and contains genetic information in the same order. Okay, so chromosome 1 and 1 are homologous. 2 and 2 are homologous. 3 and 3 are homologous, so on and so forth, right? So these two are homologous to each other. These two are homologous to each other. Okay. So if we want to do meiosis, the first thing we need to do is we need to copy all the genetic information. And we need to do DNA replication, basically. So when that happens, we're gonna have two chromosome 1 that are connected together. And then we're gonna have two chromosome 2 here that are connected together. But then the same thing for the blue one, right? The same thing for the blue one. Okay. So this is DNA replication. And again, just some terminology for you. These two right here, the red and the red, they are called sister chromatids. Okay? They are identical copies of each other, right? And they're held together at the centromere. Right? The blue and the blue here, like this blue here and that blue, those are sister chromatids as well. But the red and the blue, they are not sister chromatid, right? They're not exact copy. They are homologous. So this here and this one, they are homologous, just like before. Do we have any question in this picture so far?
No, sir.
Okay, thank you. Now, if you think back to mitosis, right? So again, like, look at my fingers, right? This is chromosome 1 from mom. This is chromosome 1 from dad, as in like your mom and your dad, right? And in mitosis, you're gonna copy them. So you get two copies connected together. These chromosomes will not find each other in mitosis, right? Chromosome 1 from mom and chromosome 1 from dad, they are like just randomly located in the cell. They're not going to find each other. However, in meiosis, which is what we're talking about, once you copy them, chromosome 1 from mom, chromosome 1 from dad, homologous, once you copy them, they will actually find each other to form this structure. Right? There are 4 pieces of chromatids here. So this whole thing, this whole set is called a tetrad. This whole set is also called a tetrad. Okay? So in meiosis, homologous chromosome pair up to form tetrad. Okay, to form tetrate. I'll choose a different color here. Purpose of tetrate. Okay. Why are we forming the tetrate? Formation of the tetrate allows you to do genetic recombination. Okay. Crossing over occurs and allow exchange of genetic information between homologous chromosome. I just copied that down first. I'll explain what it means.
Sir, your mic is muted.
Oh, sorry. Thank you. So we have exchange of genetic information between the red and the blue. A little bit of blue is going to get on the red, and a little bit of the red is going to get on the blue, and that's what we call crossing over. And now to keep it simple, I'm only doing the exchange at the ends of the chromosome. But like you should understand when this process is done, it looks more like this. Okay, it looks more like you have the red like this. Don't draw this part. Okay, just watch. Okay, you have the red, but then you would have bits and pieces of blue that are exchanged onto it, right? And it happens on both of the strand as well. Okay, both of the strand. And the same thing for the other one. It's not just totally blue now. You have patches of red on it as well. Okay, so that allows you to exchange the genetic information. Right? It's not just purely the red, purely the blue. You can mix it up a little bit. Okay, so this crossing over creates genetic diversity in gametes. Okay, all the gametes produced by a person are going to be different. Now, I know this can be a little bit confusing, so please let me know if you have any question. All right, so let's pause for a second. And I'm going to go back to the study guide. Hopefully you printed your study guide. And I'm going to go to this table on the second page. If you don't have it, that's okay. Just quickly draw a table in your notes. Put mitosis here, and then put meiosis here. Okay, and then we're gonna summarize some of the things that we've learned so far, just to keep you, you know, keep track of the progress, right? So please help me out. In mitosis, how many times does the cell divide? In the process of mitosis, how many times does the cell divide? How many times does it split? Uh-oh, that's a simple question. One division, okay? One division. You have a parent cell, and then it splits like this. Parent cell, daughter cells. So this is one division. For meiosis, the germ cells split, and then they split again. Okay? So this is two divisions. Okay? We call it meiosis. Meiosis 1 and meiosis 2. So that's the first main difference. Number of daughter cells produced. Well, you can tell from the drawing, the mitosis creates two daughter cells. And in meiosis, you can produce up to 4 daughter cells. I will explain what I mean by that later. You know what? Let's say up to 4 gametes. We'll say gametes. Right? Sperm or egg. Okay, in mitosis, are the daughter cells diploid or haploid? Do they have 2 sets of chromosomes or 1 set of chromosomes for mitosis?
One set.
1 set. Okay, thank you. Anyone else? For mitosis. Okay, so for mitosis, we are making exact copies. Okay, so they are actually 2 sets. Okay, diploid. Whereas for meiosis, they are going to be haploid, just 1 set. And this is talking about humans, right? So that would be 46 chromosomes in total and 23 for meiosis. All right. The prophase. What's the similarity and differences? Or difference here mainly in the prophase of mitosis versus meiosis. Okay, so here is the cell. Here's the cell again. So for mitosis, I'm gonna make this a little bit thinner. For the mitosis, the chromosomes after they duplicate, right? They don't find each other. They're just like, on their own. Right? They don't find each other. the big one and the big one are homologous, right? But they don't pair up. So here we'll say homologous chromosomes do not find each other. They are just randomly located in the cell. On the other hand, in meiosis, we just learned that the homologous chromosome will find each other and form the tetrad. Okay. To form tetrad. And then the purpose of forming the tetrad is to allow crossing over occurs to create genetic variation. Okay, so that's everything we've learned so far. Please let me know if you have any questions. It's only going to get more confusing. All right, let's go back to our notes and we're going to come back to this chart in a bit. Where are we? There we go. Okay, so what happens next, right? This happens in prophase, okay? Prophase 1. Because meiosis is divided into meiosis 1 and meiosis 2, we have prophase, metaphase, anaphase, telophase 1, and then we would have prophase, metaphase, anaphase, telophase 2. Right? So what happens in metaphase 1? And this thing is getting really, really big. So here we go. This is metaphase 1. So in metaphase 1, the entire tetrad is going to line up in the middle of the cell. Okay? And the order is random. Like for the first tetrad pair, it might be red on top, and then the other one could be red at the bottom. It's totally randomly arranged. Okay, let me draw a little bit better. This goes right here. And remember, they already did the crossing over. So there's going to be a little bit of the blue on the red, and then a little bit of the red on the blue, right? Okay, because of the crossing over. Right? You can put them on any way you want. Here, just add a little bit blue here, a little bit of red there. And of course, we have these little things that are at the end of the cell. You might remember them from lecture 3. Right? Those are the centriole, right? Centrioles. And the purpose of the centrioles, if you recall, is that they will send out these spindle fibers, right? To help guide the chromosome movement. Okay. So here, tetrad lines up at the center of the cell. Remember, we are only looking at 2 chromosomes here. In total, there will be 23 tetrads that are doing this, right? All lining up in the middle. And then what happens is you will have the anaphase. Anaphase. Anaphase is going to pull the homologous chromosomes apart. And remember, you already did the crossing over. So there's a little bit of red on the blue and a bit of the blue on the red. Okay. So this is anaphase. Where homologous chromosomes separate. And move towards the opposite ends of the cell. And then finally, we are going to have our, see how I can do this. There, we're going to have our cytokinesis and the telophase. So look, right? Like these two will end up in one cell, and then these two will end up in another cell, right? It's gonna look like this. Do you guys understand?
Yes, sir.
Thank you, Roman. Okay, so let's count our chromosome again. At the beginning here, right? This is 2n. I'm gonna put the chromosome number here for you to see, okay? 4 chromosomes. Of course, like again, right? Like in total, it's 46, right? Okay. So over here, what's going on here? After DNA replication, it is still 2n. It is still 4 chromosomes, right? If they're stuck together, it's counted as 1. I keep on saying that. Okay, even though we have 8 chromatids, right? But there's still just 4 chromosomes, 2 full sets. And then over here, same thing, still 2 full sets. Over here, still same thing, no changes. But then over here, once they divide, how many chromosomes do we have now? We only have 2 chromosomes. right? And only one set, which means we are haploid now. Right? One chromosome 1 and one chromosome 2. Right? That's why it's called haploid. One chromosome 1, one chromosome 2. If they're stuck together, it's just one. So we have one N here. This is also one N, 2 chromosomes. Okay. Now, I'm not gonna redraw the whole thing, but meiosis 2 is just same as mitosis. Okay, these things are gonna these chromosomes are gonna line up again in the center, and then you pull them apart again, and then in the end you're gonna get this. Okay, take a look and see if this make any sense to you. This is meiosis 2. Okay, you're gonna go through the same phase: prophase 2, metaphase 2, anaphase 2, and then okay, you're gonna get 4 cells. You're gonna get 4 cells, and look, focus on this for a second. Okay, like this one, it's gonna separate, right? Each half is gonna go to one cell. So you're gonna have one cell that has the long red one, the one on the left, and then the other cell is gonna have the red one and a bit of the blue. Right. Same thing for this one. It's gonna separate. One cell is gonna get the long blue, and then the other is gonna get the long blue with a bit of red. Right. Same thing for the short one, for chromosome number 2. Right. One cell is gonna get the short one, maybe with a bit of red. Oops, with a bit of red. And then the other one is gonna be entirely blue. And this is all random. Okay, which one you get is completely random. Like that. Right? So you would get 4 genetically different gametes. That's all. Okay. Now, of course, I'm not going to ask you to draw anything on the test, right? But understanding this process is going to, help you do some of the questions. Okay, I want to go back to the table and finish it. Okay, so metaphase 1, metaphase versus metaphase 1. There we go. In mitosis, the chromosome just kind of all lined up in the middle by themselves. Okay, of course, you have your centrioles at the end. Okay. Individual chromosome lines up in the middle. Whereas for meiosis rather, sorry, they all sound the same. It is the tetrad that lines up. Okay. The crossing over already happened, okay? But then I don't want to make the diagram too complicated. Just in your mind, make a mental note that the blue has a bit of red and the red has a bit of blue in it. Okay, so this one, tetrad lines up in the middle. Okay. So I'll give you a moment to copy that. then we'll finish the chart. All right, genetic relationship with the parent cell for mitosis is genetically identical. For meiosis, it is genetically different. Not only are they different to the parent, they are also different with each other. Right? Gamete 1 is not same as gamete 2. No two sperms are the same. No two eggs are the same throughout your entire lifetime. Yeah, the chances of creating two exactly identical gametes in your lifetime is virtually not possible. Right? The chances are so small. Type of cells involved, somatic cells. And these are germ cells, right? Germ cells. Why do cells do mitosis? For growth and development, for repair, and sometimes for asexual reproduction, right? But we're talking, this chart, this table is talking about humans specifically, so I'm not going to put that. And for this, why do we do meiosis?
Genetic diversity.
That's right, genetic diversity and gamete production. Very good. Thank you. There is the table, okay? That compares it. Hopefully it makes sense. All right, let's go back to the PowerPoint slides. This is basically everything I told you. You can go through the slides yourself. That shows you the meiosis, but we drew it already. You probably would have a, you can just read through it and compare what we drew and see if it makes sense. Okay, comparing mitosis meiosis, we did that already just now. Here's a table that have a more in-depth comparison. You can also use that as a reference. So now let's take a closer look at the actual difference in male and female. Right? So meiosis is also called, actually they're not really the same, but gametogenesis. Genesis is to create, right? So this process, gametogenesis, is to create gametes. So how do we create gametes? We do meiosis, right? So it's kind of similar. So gametes are cells involved in sexual reproduction, the sperm and the egg. You should know that by now. Where does gametogenesis occur? Takes place in the gonads, the reproductive organs, testes for male and ovaries for females. For male, it's called spermatogenesis. Spermatogenesis. That is the production of sperm. And it's just normal, regular meiosis, exactly what we have learned in the past hour. For female, it's called oogenesis. That's how you pronounce this, oogenesis. And that is the production of egg or ovum. Ovum is a fancy word for egg. That's why it's called oogenesis. And it's complex meiosis. It's different because it's different compared to the male in several ways, which I'm gonna show you. All right. So on the left here we have spermatogenesis. And you start with 46 chromosomes. And then after the first division, how many chromosomes do we have? I crossed it out here because I want to ask you. What number should I put in here? Should I put 46? Should I put 23? Should I put 92? What should I put? Come on. Show me you were with me this whole time. 46. Okay, anyone else?
23.
23. All right. Anyone else? Anyone wants to go with the 46? Anyone want to go with the 23? Let me know. Or type your
46. 46.
No, you change your mind. Okay. Maybe don't change your mind just yet. Anyone else? The rest of you here, Kiana 46. Okay, let's take a look here at our beautiful drawing. Where is the notes? Right here. Okay. So look, we started with 2N, right? 2 sets, 4 chromosomes in our example here. After meiosis 1, look, after meiosis 1, it's just down to 1N, right? It's just half the chromosome already. You see that? Right? We started with 1, 2, 3, 4. After meiosis 1, it's only 1 and 2. Right? This whole thing counts as 1, right? So 1, this whole thing counts as 1. So 1 and 2. Right? So if we started with 46, after meiosis 1, it is just going to be 23. Okay? This is 2N, this is N, this is N. Are we okay? All right, so then this is meiosis 1, meiosis 2, and then you end up, if you're a guy, then you end up having 4 sperms. Okay, fantastic. Over here, we have oogenesis. Okay? So by the way, like this spermatocyte and the oocyte, that's the name of the germ cell. That's the name of the germ cell. Okay, you don't really have to know that. So for female, after the first meiosis, again, 23 and 23, I hope you know that now. Now, it's a little bit different here for the male and the female because the egg is really big, right, compared to the sperm. Like there was a picture of it earlier, right? Like a tiny picture here. Look, this whole black thing is the egg, and then the sperm is tiny. Right? If the egg is like a basketball, then the sperm is like it's like a bean sprout, right? I don't know if you can visualize that. That's the only thing I can I can think of. It's really small, okay, basically. So the point is when female produces the egg, they don't want to do equal splitting of the cytoplasm. Okay, instead you have uneven cytokinesis. Cytokinesis is the division of the cytoplasm, right? You should know that because you did a lab for me and then you took a quiz. So when it's split, one cell gets most of the cytoplasm, the other one just get a tiny bit. Okay? So the one that didn't get as much cytoplasm, that is called the polar body. Okay? The polar body is not going to become an egg. Okay, sometimes the polar body will split again and make more polar bodies, but these are just going to be broken down or reabsorbed by the body. It's not going to be used basically. So that's why when we fill in the table, if you think back 10 minutes ago, I said meiosis can produce up to 4 gametes. In guys, 4 sperms, but in female, just 1 egg plus 3 polar bodies, basically. Okay, so if we go back to this thing, I'm always in the way. If we go back to this, we can make a note here. Okay, we can make a note. Say, male, it is 4 sperms, and female, it is 1 egg plus 3 polar bodies. Okay, it's sometimes you just get one polar body, like the polar body that you get from the first time doesn't divide again, right? Then you just have one, but doesn't matter. The polar bodies, it's not gonna do anything anyways. Are we together? Okay, let's go back to here. Now there are a couple other differences that we should point out. For example, in spermatogenesis, obviously it occurs in testes. Oogenesis happens in ovaries. We know that already. I also talk about how there are 4 sperms, but only 1 egg in oogenesis. Another difference is this: spermatogenesis is a continuous process. Right? Once the male reaches puberty, okay, they would continue to produce sperm. And the testes can produce up to 200 to 300 million sperms every day. If you do the math, that's like 2,000 to 3,000 sperm per second. It's like a continuous process. It doesn't stop. But about only about half is going to be viable. But still, that's a lot, right? That's a lot. And this process continues to, quite old in old individuals. Like you hear in the news, right? Sometimes celebrities, like they can have kids when they're like in their 80s or something like that, right? It's like ridiculous. Now, on the other hand, the oogenesis is a discontinuous process. Okay, it turns out the female, even before they are born, the process of meiosis already begins. Okay, so before birth, while the baby girl is still inside the mother's womb, the oocyte, that's the germ cell, right, is going to start the process of meiosis, and it's going to stop after prophase 1. Okay, so you have some exchange of genetic information, and that's going to stay there, suspended in prophase 1 until puberty. Okay? Once puberty occurs every month, one egg will be chosen, and it will continue to do the meiosis. Okay? It will go from prophase 1 all the way to metaphase 2. Okay? And then it will stay there, it will stop there. And then the process will finish when fertilization occurs. Okay? So the female are born with all the eggs they are ever going to have in their life. Okay? So the quality of those eggs tend to deteriorate with age, right? And that's why people who have kids at an older age, it's associated with a higher chance of genetic disorders, okay? Because mutations could have accumulated over time, and the quality of the egg is going to degrade as well. Okay, so that's spermatogenesis and oogenesis. Let's take a quick break, 5 minutes. When we come back, we'll talk about what happens when things goes wrong, okay, in meiosis. Okay, so see you in a bit. All right, let's keep going. Let's take a look at what happens when we have some kind of mistakes in meiosis. And one of the things that could happen is what we call non-disjunction. In non-disjunction, the chromosome fails to separate when they are supposed to. Okay, so in meiosis, there are 2 places that could happen. It could happen in anaphase 1 or anaphase 2. And depending on whether it happens in meiosis 1 or meiosis 2, the results are going to be a little bit different. So let's take a look at it visually, okay, rather than just reading the words. To keep it simple, we are just going to be focusing on one pair of chromosome. Okay, so this is just 2 chromosomes. Okay, and this is 2N. Okay, So if you have non-disjunction happening in the in meiosis 1, what happens is like these two things are supposed to separate from each other, right? One goes to each end, but if they don't separate, then the entire thing will end up in one cell, and then the other cell will be missing chromosomes, right? Again, like I know this is empty, but remember, in a real cell, you have the entire 23 pairs, right? So like you basically will be missing a pair. So if this cell goes on to do meiosis 2 and nothing is wrong in the second division, then they will separate. And you will end up having 2 cells, but each cell will have an extra chromosome. If this cell goes on and do meiosis 2, you will end up having 2 cells that have 1 less chromosome. So in this case, where the non-disjunction occurs in meiosis 1, you end up having 2 gametes that have extra chromosomes, and we have 2 gametes with missing chromosomes. Okay, so that's pretty bad. Over here, if we have normal separation in meiosis 1, then that's fine. You know, they separate as they were supposed to. And then what happens is, if non-disjunction happens in one of the cells in meiosis 2, then you end up having one cell with extra chromosome and the other is missing. Okay, and then these two are just fine. So non-disjunction is worse when it happens in meiosis 1, because all the gametes are going to be is going to be problematic. If it only happens in meiosis 2, right, at least you will get 2 of them that are normal. Okay. So what happens if these gametes go on and fertilize and get fertilized by a sperm? Well, in that case, the baby is going to end up having what we call trisomy. Tri means 3, right? So instead of having 2 sets of chromosome, they will have an extra chromosome for whichever one that was not separated. So like if chromosome 20 was not separated and it made it to the egg, then the person will be born with trisomy 20, right? 3 chromosome 20. Trisomy 21, that would be 3 chromosome 21. Right? So it's 2 full sets plus an additional chromosome. Okay. So that is common for many diseases that the baby can be born with. But if we have fertilization of gametes with missing chromosome, like one of these ones, the empty ones, the 22 here, even those get fertilized, the pregnancy is not going to be viable, okay, because it's usually lethal. So how do we know? Well, it's possible to check the DNA of the baby if you know, if parents are want to find out, or if there's a family history of these genetic disorders, and you know, or if the family is at high risk of having some of these genetic abnormalities. Remember, I told you the eggs, the quality is going to degrade as the person age, right? So if the parents are a little bit older, if the mom is a little bit older when they become pregnant, they might choose to screen for some of these genetic abnormalities. And to look at all the genetic material, we can do what's called a karyotype, right? We learned that previously. We line up all the chromosomes like this, and it gives a very clear visual a way to see if there are any extra chromosomes, right? So if you look here, this is a normal karyotype. Right? We have chromosome 1, 2, 3, 4, right? Each one of them is just a pair. There's no extra, there's no missing, right? So some genetic disorder, like Down syndrome, that is extra chromosome 21. And in some other cases, people might only have 1 X. So I know I told you, right? Like missing chromosome is usually lethal, but when it comes to sex chromosome, it turns out is okay, right? I mean, there are going to be problems associated with the disorders, but like it's not lethal. Okay, so monosomy means only one chromosome X. And so how do you look at the baby's cells? It turns out there are two methods to do this. The first one is called amniocentesis. Amniocentesis. So the baby, when it's growing in the mother, is actually surrounded by these fluid called the amniotic fluid. Before a woman give birth, like before a woman goes into labor, they say the water breaks, right? And then all this liquid comes out. The liquid is the amniotic fluid, basically. Right? So that fluid contains a bit of the baby's cells, and so what we can do is what the doctor can do is they could take a sample of the fluid, extract the cells, and look at the DNA. So that involves injecting a needle just at the belly button. And the whole process is guided by ultrasound, so we don't want to be poking at the baby, right? So you take the fluid out, prepare the cell, and here's the karyotype. And you can see in this particular case, there is an extra copy of chromosome 21, which means the baby has trisomy 21, Down syndrome. So this can be done between 15 to 20 weeks of pregnancy, so still relatively early on in the pregnancy. Whole pregnancy is 36 to 40 weeks, right? And that's an option. Now, if people want to screen even earlier, there's an other option called CVS. CVS stands for chorionic villus sampling. The chorionic villus is a tissue that is connected to the placenta, and it contains the same cells that the baby has. Right, so you just cut a little bit of biopsy, a little bit of tissue from there, and then you will have plenty of cells to examine. This can be done even earlier, from 9th week to 14 weeks of pregnancy, and parents can make an informed decision as to what their next step is. If they find out there are certain genetic disorder with their babies, right? So CVS or amniocentesis. Okay, let's do some poll questions and see if you can do this. Okay. After meiosis 1, the cell is considered as anyone else? Okay, I only got 3 answers, but they're all incorrect, unfortunately. Okay, after meiosis 1, the cell is already haploid. Okay, we roll it down here. You see? After meiosis 1. Okay, we started with 2n, and after you do all the division, is going to be n. Okay, it's already half the genetic material. Okay, so that's the answer. It's going to be haploid. Next question. Since you guys are not really participating, I'm just going to tell you the answer. Which of the following is true regarding human gametes? Okay, gametes are sperm or egg. It is produced by the process of mitosis. That's not true. Gametes are produced by meiosis. Contains 23 chromosomes in gametes, not 46. Produced in testis and oviduct. It's ovaries. Oviduct is like the fallopian tube, right? That's where fertilization occurs. All are haploid. That's the correct one. Which of the following is true for an organism that has chromosome number of 2n equals to 16? 2n equals 16 means n equals to 8. So one set has 8 chromosomes. That's what this means, right? One set has 8 chromosomes. So let's see. The organism has 16 sets of chromosomes per cell. Nope, it has 2 sets. That's why it's 2n. A gamete from this organism has 4 chromosomes. Is it not true? If total is 16, then the gametes are gonna have half of that, right? So half of 16 is 8. The organism is sterile. That's not true. Sterile means odd number of chromosome sets, right? So it has 2n is not sterile. Okay, only for odd number sets. Each cell has 8 pair of homologous chromosomes. That's true. One set has 8, and then it has 2 sets, right? So 8 pairs. That's the correct one. Organism is diploid with 32 chromosomes per cell. Nope. 16 chromosomes per cell. It says right here. Right? Okay, moving on. Now we're going to talk about the molecular basis of heredity. Okay, and this here is Gregor Mendel. He's credited as the father of genetics, okay, because of his work on pea plants and discovering how certain traits are passed on to the next generation. So some terminologies to refresh your memory. A gene is a section of DNA that encodes for instruction to make the protein. You should know that by now, right? We did a lot of those things, right? Transcription, translation. But we also mentioned how genes have different versions. We call them alleles. And the example I keep on using is eye color. Okay, blue eyes, brown eyes, both are for eye colors, but they are different versions. So different alleles. We are diploid, which means at most we will have 2 different alleles. They don't have to be different. They could be the same, depending on who your parents are, right? If they are the same allele, then we say that we are homozygous or pure for that allele. If they are different, then we say it's heterozygous or hybrid. In chemistry, you learn about homogeneous and heterogeneous mixture, right? Homogeneous means it's the same throughout, like salt water. Heterogeneous means it's different, right? Like oil and water mixed together, right? So in the context of what we're talking about, it's the same. Homo is the same allele. Hetero is different allele. Let me give you a concrete example. We have 2 chromosomes here, the red one and the blue one. And for the last time, can someone please tell me again what's the relationship between them? If I have to use one word to describe the relationship between the pink one and the blue one, what is the word I'm looking for, guys?
Homologous.
Homologous, that's right. They're not identical. They will only be identical if after, you copy them in DNA replication. Okay, so this is homologous. They are same length, same size, and they contain genetic information in the same order. You see it's A, B, C, D, E here. It's also A, B, C, D, E here. It's not going to be, for example, it's not going to be A, B, C, D, E on one and then A, C, D, B, E on the other. It has to be the same order of genetic information. So we use letters to represent the gene. Okay, and you can see the ones that I'm circling, they are different, right? Big A, small a, big B, small b, right? That's another one here. That's another one. So anything that's different, We call them heterogeneous. Oh no, sorry, that was chemistry. Heterozygous. Okay. Heterozygous or hybrid. Okay. On the other hand, if they are the same, small d, big E, small h, small l, and I. Okay, these ones, all the ones that are the same, we call them homozygous or pure. Okay, pure means the same. Heterozygous means different. You could be heterozygous for one and homozygous for the other one. There are no relationship. Whether you're homozygous for one trait or not has no effect on whether you're homozygous on the other one. Okay, it all depends which sperm fertilized which egg when you were conceived. Okay, it's all by chance. Okay, all randomly selected. So a couple of other things here, other terms. Okay, a character is a heritable feature, something that can be passed on to the next generation, right? Such as flower color. In the case of humans, right, it would be things like eye color, my favorite example, right? As you know by now, because it's the easiest to visualize. Not everything about you can be passed on to the next generation. If you go to the gym a lot and you're very muscular, very big and bulky, your kid is not necessarily going to be grow up muscular, right? Because that's not a heritable feature. Is an acquired trait, right? Something that you gain throughout your life is not going to be passed on. Okay? If you dye your hair purple, your baby is not going to be born with purple hair, right? So not everything is going to be heritable. For things that are heritable, we call that a character. So blood type, eye color, whether your earlobe is attached or detached, so on and so forth. A trait is a variant of the character. Okay, why do we have different traits? Right? That's because we have different alleles, right? Different allele is responsible for the variation in the character. Now, some traits are dominant while some are recessive. We use capital letter to represent dominant trait, and we use lowercase letter to represent recessive trait. So I'm going to give you an example to help you understand this. All right. Again, just use eye color. Big B is brown eyes. Okay, and small b is blue eyes. And we use the same letter for the same character. Okay, use the same letter for the same character. Okay, the character in this case is eye color. You cannot use big B for brown eyes and then letter C for blue eyes or something. It has to be the same letter. The big letter, the capital letter, is the dominant trait, and then the little letter, the lowercase, is the recessive trait. So I'm gonna ask you this. Okay, it's just tell me. Please tell me. Okay, please tell me so I know you understand. If someone is big B, we say homozygous, right? Because it's the same letter, same case, right? Capital letters. What color is the person's eye color?
Brown.
Brown. That's right. Okay. What about small b?
Blue.
Blue. Okay. Thank you. The rest of you understand? Anybody not understand why it is brown and blue? Speak up now, because it's only going to get more confusing later on. Okay, what about big B and small b? Now someone is heterozygous. They are hybrid, big B, small b. What color do you think the eye color is going to be? Is it brown? Is it blue? What's going on?
Since brown is more dominant,
Mhm.
it's still gonna be brown.
That's right. Okay. So because brown is dominant, it doesn't matter you have a blue-eye gene, it will not show. Right? That's what dominant means. Okay. Dominant means it will always show. Even if you just have one, it will show up. Recessive means it will only show up if the dominant is not there. Okay. So if you see someone with brown eyes, you don't know if they are big B, or if they're big B, small b. You don't know. You have to figure out their family background. But if someone have blue eyes, then you know they are small b. Okay. So that's what dominant and recessive means. Now here is a bunch of dominant trait in humans and a bunch of recessive traits. Okay. So for example, like freckles. Right? Freckles are like the little dots here that some people have. If you don't have it, then that's recessive. Okay. If you take a look at hair, actually, you know what? Let's find something else. Let's find the where's the earlobe? I thought I saw the earlobe. Oh, there it is. It's the next one. Okay, earlobe. Okay, suspended or attached to the head. Okay, like if you look at your earlobe, right? Like if you have that little thing there, that's called suspended. But if you have these kind of earlobes, okay, then it's called attached to the head. That's what it means. Like so, like you can feel your earlobes and you can see, right, whether you are dominant or recessive. Dimples, right? Dimples. Right? Do you have a dimple or do you not have a dimple? Right. So some traits are dominant and some traits are recessive. Here is the thing to note, though. Okay. Let's say we look at no dimples. Okay. If the mother and the father both have no dimples, okay, so little d, no dimple, right? What can you say about the kids? Are the kids gonna have dimples or no dimples?
The kids won't have dimples.
Will not, okay. Anyone else? Anyone else? What do you think? Mother is no dimple, so little d for the recessive trait. Father little d. Whole family is gonna be no dimples, right? Because the mother can only pass on the little d and the father can only pass on the little d. Right? So if the parents both have a recessive trait, then their kids are going to have a recessive trait as well. Make sense? Now here is another thing. This is called polydactyly. Some people are born with 6 fingers. Right? 6 finger is dominant trait. Right? So if you don't like your kids to have 6 fingers, and you have 5 fingers, all you have to do is find someone else who also have 5 fingers, right? And then they will guarantee your kids is gonna have 5 fingers, right? Unless they cut it off before they met you, and then like you don't know, and then there's a chance. It's actually rare for people to have the full 6 fingers. In many cases, it's just a little appendage on the side. So some people do actually cut it off because it's like non-functional. It's kind of getting into the way, and it doesn't have to happen in the hand. It can happen in the toes as well. Okay, so that's a list of all the traits that are dominant and recessive in humans. It's interesting to look at. All right. Couple more terms that I want to throw at you. Genotype. Genotype is the genetic makeup of the organism, of the person, of the plant, whatever we are looking at, right? In other words, what kind of allele do they have? In light of what we talked about, you know, big B, small b, small b, small b. These are all genotypes. Okay, the phenotype is the outcome, the expression of the genotype. What do they look like? Is it brown eyes or blue eyes? right? That's the phenotype. So as you saw, two people with the same phenotype can have different genotypes. Okay, two people who have brown eyes, one could be homozygous and the other could be heterozygous. You just don't know. Okay, so in this case, you're both. We use curly hair, and if you look at curly hair, curly is dominant, right? Curly is dominant. So if two people who are both of them have curly hair, you know, one could be big C, right? The other could be big C, small c. You can't tell unless you check their family background. Okay. So let's take a look at Mendel's experiment. He basically experimented with peat plants. Peat plants are like, the peats that we buy from supermarket, okay? And the reason he did it is because it's easy to grow the plants, and you can fertilize the plant easily yourself. Like you can make this plant cross with this plant and make baby plants basically, and basically just use a paintbrush to pollinate them, right? So it's easy to set up, easy to study, and you can see the generations growing very quickly. Okay, so here are some traits, right? The shape of the seed, round is dominant over wrinkle, yellow is dominant over green, okay? And the shape of the pod and color of the pod and the flowers, position of the flower and the length of the stem, so on and so forth. So many traits. You don't have to memorize this, okay? I will tell you in the question. But I just want to show you, right? Plants are just like humans. They have certain characters, and the character have traits. Some are dominant, some are recessive. So Mendel has a law that he figured out. The first law is called the law of segregation. It sounds really like confusing, the term, but all that means is that any parent is only able to pass on one of their two alleles. If we are talking about diploid organism, okay, which we are. All right, so if you have big B and small b, you cannot give both to your kids. Okay? You can either pass on the big B or the small b. Right? 50% of your eggs will carry the big B, 50% of your egg will carry the small b. Okay. If you are big B, then 100% of your egg or sperm are gonna be carrying the big B. Okay, so law of segregation just means you have two sets of chromosomes, but you can only give one to your kids. We know that already, because in meiosis, you produce gametes that are haploid. Right? You reduce your genetic material by half. Okay. So here we have we're going to be solving problems called monohybrid cross. We're going to do that together. P generation. P means parent. Okay. Started with plants with pure round seed and seeds that are pure wrinkle. Okay, so here is a round seed. Here is a wrinkle seed. Because it's pure, pure means homozygous, right? The round seed is big R, and the wrinkle seed is little r. Okay. So this plant here, when it makes the gamete, it only have one choice, right? When the big R and the big R separates, right? 50% of the gametes will be big R. The other 50% will also be big R because there's no other choice, right? It's just big R from this plant. Right? So the gametes are big R and then big R. For the wrinkle guy, it is going to be little r and little r, right? So if this gamete and this gamete fertilize each other, the offspring is going to be big R, small r. Right? If this gamete and this gamete comes together, it's also going to be big R, small r. In fact, if you do all the possible combination, the first generation, F1 generation, they will be 100% big R, small r, which means they will be 100% round. Okay, so this is the genotype, and this is the phenotype. Genotype is what's the genetic makeup. Phenotype is what they look like. Okay. So in this case, if you take a pure round and you cross it with a pure wrinkle, all the kids will be round. Okay. Now let's do one more generation. Okay, and this is another advantage of breeding plants, right? You can take the offsprings and you can breed them. You can't do that for like animals and like humans, right? That's like incest, right? It's not ethical, right? So here, we're gonna take two individuals from F1 and we're gonna pollinate themselves. So these are F1 individuals. Okay. So let's go back and see. All the F1s are big R, small r, right? So over here, this is big R, small r, big R, small r. So if we look at this particular plant, now it has a big R and a small r. According to the law of segregation, half the gametes would be big R, the other half is going to be small r. So we put it in the table like this. Big R, small r here for one of them, and then we will put big R, small r in the other one. If this gamete and this gamete join together, we will get big R, big R. If this one and this one comes together, what are we going to get? Hopefully, in your mind, you're saying big R, small r. If this one and this one come together, also big R, small r. We put the capital letter first all the time, always. And then finally, if this one and this one comes together, we're going to get a little r and a little r. All right? So let's take a look at the genotype. Genotype, it's going to be 25% big R, big R. 1 out of 4 boxes is big R, right? How many percent for big R, small r? How many percent for heterozygous? 50% is correct. 2 out of the 4 boxes. And lastly, 25% for small r, small r. Now, phenotype is a little bit different. They are not always the same answer. Big R is round. Big R, small r is also round. So together, 75% chance that it's going to be round. 25% chance is going to be wrinkle. Any question? Okay. Let's do an example here. I'll give you a moment to try yourself. I will take it up. I'll be right back. You try this question. So for a question like this, we have to, come up with the variable, if you would. It's kind of like math, right? So it says here, red flower is dominant over white flower. Okay. So what should be our capital letter? Should the capital letter be R or should the capital letter be W? Yeah, R is a good choice because red, right? Red, right? So we'll say big R is red allele. And what would be the white allele? Little r. We're not going to use W because we have to use the same letter for the same character. Okay? Homozygous red flower. Homozygous means the same. Red is big R. So this has to be big R, big R. Homozygous white. Homozygous means the same. And white is little r. So this is little r, little r. Heterozygous means different. And this is red flower. So one big R, one small r. What about heterozygous white? Anybody have an answer for heterozygous white? Okay, heterozygous means different. Okay, and then white. Well, there's only one way to be heterozygous, right? Big R, small r. But big R, small r is red, right? So it's impossible. Impossible. Right? So if you see on a test question, it says, a white flower is crossed with whatever, and you are in a position where you're thinking, oh, it's white. Is it homozygous or is it heterozygous? It doesn't matter because since white is recessive, recessive is always homozygous. Okay. Is always homozygous. It's always pure. Okay? It's only the dominant trait that you don't know, that you're not sure if it's pure or hybrid. I think we have time to do one more examples, if you guys are up for it. Consider a type of orchid where red flower allele, big R, is dominant over white flower allele, small r. Predict the phenotype and the genotype of the F1 generation. Okay. So over here, hybrid red. Hybrid red is big R, small r. White. Again, I don't have to tell you if it's homozygous or heterozygous. White is pure for sure because it's recessive, right? We can make our own chart. It's called Punnett square, by the way. Punnett square. So we take this here, and then we can put the gametes. Half of the gamete with big R, the other half is going to be small r. Put the gametes here, small r, small r. A Punnett square. P-U-N-E-T-T-E. Punnett square. All right. So you do the combination. This and this together is going to give me big R, small r, big R, small r, small r, small r, small r, small r. Okay, you just mix and match, right? Okay. Put this here, so on and so forth. Okay. So genotype. 50% big R, small r, 50% small r, small r. And for phenotype, 50% red, 50% white. Okay. Just quickly do one more. Pure red with white. Okay, pure red with white. I'll let you try it yourself, and then I'll take it up. Okay, pure red is big R, big R. White is small r, small r. We are gonna do our table again. And we are going to have big R here on the side. Small r here at the top. And if we do the combinations, and just go across and down, right? You will get 100% big R, and 100% red. Okay, that's it. Now in the study guide that I post, there are a bunch of problems that you can try. Right here, study guide. Over here, these are all monohybrid problems. Okay, I'll post the answer as well later on. But there are 6 questions that you can try. Next class, we will learn the rest. Okay, those are a little bit more challenging. So please come. Dihybrid cross, incomplete, codominance problems, sex-linked problems, and then we're also going to do pedigrees. Hopefully we can finish everything next class. And that's it for today. All right. No quizzes this week. I don't think there's any quizzes next week either. So just take this time to rest a little bit. And I'll see you on Thursday. Thank you, guys. Have a good day.
See you, sir.
See ya.